Today we're going to, yes?
Sorry, I just have a question. So, like, if we're not going to be able to come for, like, this class period at 1:30, can we come to the one before this one today?
Yes, yeah, that's fine. Just let me know and make sure it's okay. But yeah, should be fine. Okay, so we are moving into cells. So up until now, we haven't really been dealing with living things yet. We've been doing molecules and atoms and chemistry and things which are important for living things, but now we're actually getting into our basic fundamental unit of life, which is the cell. So there are a few historical figures in biology which were important for this lecture. Who were Schleiden and Schwann? Right, so they came up with this idea of cell theory. Okay, and there were three tenets or three main points of cell theory. And what were they? I want you to talk to your neighbor and see if you can come up with all three of them, or at least bits and pieces of them. Or you can look at your notes. Okay, so what was the first point? All right, so all living organisms are made of cells, okay? And then that includes processes of metabolism and gaining energy. All right, what was the second part?
Cells are the smallest living things.
Okay, good. So this is our fundamental unit. It's kind of reiterating the same thing, but there's nothing smaller than a cell. All right, and what's the third point? All cells come from other cells, right? Okay, good. All right, so I know we talked about this before, but where does that leave us with viruses? They are not living according to cell theory, right? Which of these three does it violate?
Cells are the smallest living things.
Okay? All of them, actually. Yeah. All living organisms are made of cells. Okay? Viruses are not made of cells. Cells are the smallest living things. Okay? It's smaller than a cell. And they come from other cells. Viruses don't come from other cells. They come from other viruses. But okay, so based on cell theory, those are not living things. All right, what about Robert Hooke? Why was he important?
The first guy who actually saw cell basically.
Right, so he discovered cells. Okay, he was kind of the first person to really look at, describe, and say, all right, this is a cell. Okay, and how did he do that? Yeah, with the microscope, right? Cells are too small to see. Well, most cells are too small to see with the naked eye. But with the advent of lenses, and there were a number of different people who came up with similar things to look at microorganisms, but he was the first to look at them and describe cells. Okay, so we have 4 different types of microscopes, 2 of which you have already used in lab, which help us to have a greater resolution on the things that we can see and observe. So the first is called what's that? Okay. A dissecting microscope. Okay, and this just increases the magnification by a few times. Okay, so just really, if we want to see something that we can see with the naked eye, but we want to see a little more detail, we can put it under a dissecting microscope. Okay, the second one is the compound light microscope, where you have two lenses of which compound the magnification, and then the light from below illuminates the specimen. Okay, and so you use both of those in lab already. All right, then we have two types of electron microscopes. A transmission electron microscope, and it does exactly what it's named for. So it can actually go inside of cells, and you can form images internal to cells. You can see the insides. The other type, the scanning electron microscope, also does as described. It provides a great amount of detail, but on the surface of whatever it's looking at. Okay, so here we have 4 images. You and your neighbor match up the images to the different microscopes. Okay, and there's one of each, so I'm not trying to be tricky. Oh, okay. So you have to have the Explain Everything app. And it's just a... Okay, so hopefully this is what you got. Okay? Never leave a brother hanging. He was drunk. Okay. So again, you have, this is the inside of a cell. So you can see the internal structures. Here you can see great resolution on the fly's head or whatever that is. This is just a magnified, piece of an insect, but it's just magnified a few times. And here you have a follicle in an ovary, so you can see a lot more detail in each. Okay, good. All right, so I've got this video here, which we will watch. Okay, or you can watch on your own. Okay, and then tell me what you think, what microscope was used to make these images. This one's gross. Yeah, they're all the same. Okay, so what microscopes use for those? Good, yeah. So they come up with some pretty cool looking stuff, but it's all surface. Yes. I'm not sure if they go in and like color it later. I'm not sure. I've seen a lot of them which are black and white, but they look pretty similar. So I think they probably enhance them with color. Okay, so those are microscopes. Here are some of the things that we can see with microscopes. Our prokaryotes, okay? So this is one of two different types of cells. We have prokaryotes and eukaryotes. And those are our two major divisions of life, so the very basics. Prokaryotes are single-celled. They contain two of the domains of life. Bacteria and archaea, okay, which are also sometimes referred to as eubacteria or archaea bacteria. And what are some other characteristics of prokaryotes? Okay, they are small.
Cell wall instead of membrane.
Okay, so they have a cell wall. Yep.
Don't they asexually reproduce?
Yes. They have, yeah, they're all asexual. They have some forms of DNA transfer, but yes.
Next one, no organelles.
Okay, they have no organelles. Good. Okay. Karen?
Yeah, but it has a nuclear.
Right, good. So there's no nucleus. It has a nucleoid instead. Okay, good. That's good enough for our purposes right now. But it does share a lot of characteristics with our other types of cells, eukaryotes. One of which is, and I'm going to draw a little cell over here. It has a plasma membrane. All right, what is the plasma membrane made out of? Yeah, it's a phospholipid bilayer. Okay, good. All cells have a phospholipid bilayer, a plasma membrane. Okay, but as was mentioned before, these guys also have a cell wall. And the cell wall is made of a specific substance which differentiates it from other types of cell walls. Called peptidoglycan. Okay, you can break that apart. I know it's a big word, but you can break it apart and it tells you what it's made out of. So what is peptid? What does that refer to? Okay, peptides, right? So it's a protein, and then glyco. What does that sound like? What's that? Glycogen, right? Yeah. So it's just sugar. So it's a peptide. It's a protein and a sugar. Okay. Can with this A. Peptidoglycan. Okay. If you want to sound smart, just drop it at a party or whatever. Okay? Good. All right. Sometimes they will also have another layer, but not always. Called a capsule. And the capsule may or may not be present depending on the conditions that the prokaryote is in. Okay, so if it is too cold, favorable, or conditions are unfavorable otherwise, it will oftentimes shrink, form this thick capsule, and that'll be its protective layer, and it'll go into a dormant state and remain there until conditions are favorable again. Okay? All right, so a few other things on the outside. These little projections coming off, they kind of look like hair. These are called fimbriae. Or pili. And these are for connecting with other bacteria or aggregating with other bacteria. So they'll reach out to another bacteria, connect with their fimbriae, and stick together that way. Okay, and then they may or may not have this tail-like structure called a flagella, okay, for locomotion. Okay, so for swimming through whatever fluid it lives in. Okay, so those are all the external parts of the prokaryote. Okay, on the inside, it's going to have a circular piece of DNA called it's the chromosome. There's only one, and it is circular, but it's not usually in a circle shape. It's actually, when you look at it, it's usually all jumbled together and pieces of it kind of looks like that. But if you were to pull it apart and string it out, it would form just one complete circle. Okay? And the area that is concentrated with DNA is called the nucleoid. All right, the whole internal part of the cell is filled with a fluid called cytoplasm or cytosol. And within this fluid, it's mostly water, but you're also going to have everything that it needs to survive. Okay, so it'll have amino acids in there. It'll have bits of transcribed RNA in there. Whatever building blocks it needs to function or reproduce or make new cells or split, all of that's going to be contained within the cytoplasm. All right, and then finally it'll have free-floating, protein-making ribosomes. And again, so the ribosomes are where the proteins are made. And that's it. So pretty simple. Not much to this. When we look at the eukaryote, there's a lot more things there because there's a lot more organelles, complex membrane systems, more functions, more specificity. Yes?
So in a eukaryote, this stuff, at least ribosomes, are considered organelles. Why are they not considered organelles?
they're not membrane bound, okay? So ribosomes are made of RNA, and that's all they are, just folded RNA in a ball. But there's no membrane about them. Yes.
So they're not really there with units.
What's that?
They're not really there with units, so.
Yeah. Yeah, I mean, they're not, yeah, they aren't separated from anything. They're floating in the cytosol with everything else. So no organelles, a better way to write that might be no membrane-bound organelles would be more specific. Okay, any other questions?